Basically, we are going to start today the chapter of health and diseases, or in other words, you can say that this chapter is related to the immunity and defenses. Uh, the thing is that when we talk about the immunity or when we talk about the health and disease, first let's discuss about the paper configuration of this chapter. The paper configuration in from this chapter you'll only be getting the questions in P2 and P4, but this is a reality that this chapter includes the questions of 4 marks and 6 marks. 4 and 6 marks, both type of questions would be appearing, would be, you can say, can be asked in this question in this from this chapter. And these could be like some other questions, like 1 or 2 markers could also be asked. So keep this in mind that this is one of those chapters from which the question is almost always included. I will tell you later about some chapters which are necessarily included in the CIEs.
Okay.
Anyways, let's begin. So, what idea do you already have about this chapter?
Lack of the word
Not,
immunity.
yeah, immunity, defenses, health, disease. What do you think about
How
it?
your body copes against diseases and
Hmm,
sickness.
hmm. Basically, when we talk about the immunity, immunity is a mechanism of tackling different kinds of problems. Mechanism of tackling different invaders. That is immunity. Not only invaders, but sometimes body's own problems, and sometimes allergies, etc. So this system basically involves these particular, you can say, mechanisms. Now, about the immunity, I would say the immunity also involves defenses. When we talk about immunity, we don't only talk about the immunity that is present inside us. We also talk about the immunity or the defenses that are present outside our body. Always remember that when we talk about the defenses, we have 3 kinds of defenses in the body. First defense is called as the physical defense. And in the physical defenses, you can say our skin, which basically is impenetrable, mostly impenetrable. Remember, not always, but mostly the skin is impenetrable. Then hairs, not body hairs, it basically includes the hairs in the nose. And the third thing about the physical defenses is the cilia present in our trachea, etc. So these 3 types of defenses are physical, and they can literally trap the particles. Both of these things can trap the dust and microorganisms. Second thing, microorganisms. The second type of defense, this is the first type. The second type of defense is called as the chemical defense. The chemical defenses are those which literally include some chemicals being released on the skin or inside the body. First chemical that is being released is called as the sebum. The sebum basically is a type of chemical that could that could generally be covering the skin, leading to the you can say protective mechanisms. The second thing is the saliva. The saliva also has some antimicrobial activities, antimicrobial properties. And the third mechanism is the third mechanism is the hydrochloric acid. Hydrochloric acid. All these things are chemicals, and these basically are going to tackle organisms uh, which can be entering with the wind, with the air, and they can enter with the food as well. So these different kinds of defenses are there. A very important thing that I would also, okay, I'll mention that later. Okay, so the third defense, first I'll tell you about the third defense. And that third defense of our body is the cellular defense. What does it typically means? The cellular defense is type of defense that you can say is carried out by the white blood cells. It is carried out by the white blood cells. Now, the white blood cells actually are those which can be divided into two categories, out of which we have talked about one. One type of white blood cells is the phagocytes, and the other type of the white blood cells are the lymphocytes. Now, the white blood cells, um, how do white blood cells act? Before knowing that, I would like you to know one more thing. See, when we talk about defenses, this is a general term, but in parallel, we also talk about immunity. Immunity and defense, these two are, you can say, synonymous terms. At least at this level, these two are synonymous terms. Immunity can be divided into two categories. One type of immunity is called as innate immunity. Innate immunity is a type of immunity which we do not need to learn. It is present since the time of birth. It is present since the time of birth. So innate immunity is not going to be, we do not need to learn it. Okay. So in the innate immunity, you say it is since birth. What kind of mechanisms are involved in the innate immunity? What kind of mechanisms are basically defending us since the birth? This physical defenses are involved in innate immunity.
So
Then the
basically, like innate immunity is something that you don't build over time. It's just you're born with it.
exactly, exactly. Similarly, chemical defenses are same. They are also since birth. In the next category, when we talk about the phagocytes and lymphocytes, this immunity, these cells basically are the part of innate immunity. Innate immunity, which means that these cells are going to be present in our bodies since birth. Talking about the lymphocytes, they are present since birth, no doubt, present since birth, but they learn to adapt to tackle new organisms or new pathogens. Now, this kind of immunity is called as learned immunity.
It's the one that you build up.
Yeah,
So
learned immunity or
what example like when you get COVID, for example, the
mhm.
second time you probably build immunity for it, right?
um, you said COVID. Then what did you say after that? I could not hear you properly.
But basically, you build an immunity of it for it, right? So
Yeah.
there's a less chance of you getting it again.
Exactly, exactly. We basically are, when we talk about the COVID, when we talk about the corona, or when we talk about those diseases for which we get the vaccines, we are basically learning for those diseases. I will try to make you understand that what is the meaning of learning, by the way, but that topic is majorly the part of A level. I will try to compensate, staying within the limits of our syllabus, that how we basically learn the things. So how our immune system adapts the things. So innate immunity and adaptive immunity, this immunity is also called as acquired immunity, which we have acquired later in life. So lymphocytes are involved in that kind of activity. Phagocytes, some other white blood cells are also involved. White blood cells are of many types, but we are only going to discuss lymphocytes because we have talked about the phagocytes already. Okay, so always remember that we are possessing the lymphocytes since our birth. Now, what happens? Uh, First of all, I need to clear out a few things. See, if you get a wound, if you are hit by a splinter, your skin is being pierced by the splinter, or like if basically something is basically puncturing your skin, then what is going to happen at that time? Mostly innate immunity works, mostly innate. And even even the uh, uh, random types of pollens are also being treated. Random invaders, they are also being treated by the innate immunity. Sometimes you inhale the pollens and your body suddenly start reacting. That is mostly innate immunity. I'm not saying always, but it is mostly innate immunity. Similarly, other things are there. But when there is infection, if infection is there, infection means that the microorganisms have overcome all the defenses except lymphocytes. They have overcome all the defenses when there is infection and fever. So it means that at the site of wound, your body would be immediately your body would be, so suppose that this is a wound, this is your skin, this is your skin, and you got a splinter puncturing your skin, at that site you'll see that the blood capillaries would be punctured, the blood is accumulated. Now the from the closest capillary, the phagocytes, suppose this is a closest capillary, so the phagocytes would closest capillary, inside this capillary you would have these large phagocytes. They would enter here, they would go and they would kill microorganism, they would kill and killed. What is the meaning of it? They would kill them, and they would get killed. Have you ever seen, do you know that why the pus accumulates in our body?
No, I don't know why.
The pus accumulates when you have a wound and the phagocytes reach there, and those phagocytes are killing and getting killed. They're killing. When the phagocytes get killed, your wound starts getting whitish or pale white, which basically appear as pus. Whenever you are getting an infection and you see greenish area, you see greenish appearance, that is called as pus, and pus indicates that your wound has gone bad. Like you may have observed that when there is too much cold, um, the your nose starts running, but but the mucus that is coming out of it is whitish. It is
Yeah.
transparent. I'm sorry, it is transparent. If it is transparent, it means there is no problem. You are only getting cold and your nose is was getting dried. But if that runny nose is producing green material, that is when you need to worry, because in that case you have been infected. So so it means that you are typically your white blood cells are typically getting killed and they would appear green. So much white blood cells are killed now that they would appear green under your skin or inside your body. But suppose that there is a tissue. Let's say there is a disease that you would study about, but let's say the organ is liver tissue, and it is infected, and maybe due to bacteria, maybe due to bacteria or virus, due to bacteria or virus. Would you excuse me for 5 seconds?
Yeah.
So sorry. So basically, if a liver tissue or any other tissue has been infected by the bacteria or virus, what's going to happen that first of all, first thing, now please be very careful. Now I'm going to draw a map and I'll explain it verbally. You have to be very careful while listening, and basically every each and every step would be described verbally, so you can listen to the recording again. I may not be writing the points, but I'll explain everything verbally. So
Okay.
suppose that this is the site of infection. First of all, your phagocytes would try to reach over there. The phagocytes, they would try to reach over there. Who would call them? Who would call them? Remember that these infected cells, these cells are basically infected and dying. These are infected and dying cells. So they would release some chemical signals that would reach the phagocytes, and phagocytes would try to reach at the site of the infection. So this is the first step. That after a proper signal, the phagocyte would try to reach this infection site, trying to kill the organisms that are infecting our organs. But it is insufficient because phagocytes are dying. Instead of controlling the infections, they are dying. So it means that the infection has now overcome the innate immune system. Now, what's going to happen? These chemical signals are do not limit themselves to the phagocytes. They also reach to some other cells which are called as the lymphocytes. Okay, I'll try to tell you over here. Lymphocytes are of two types: T lymphocytes and B lymphocytes. T and B lymphocytes. So the first of all, these signals would reach to T lymphocytes most of the time. These signals would reach to T lymphocytes. The T lymphocytes would immediately move to the site of infection, where they would now they are going to do two things. These are the T lymphocytes. They are going to do two things now over here. First is they would immediately release signals signals to call upon the B lymphocytes. Immediately they would release the signals. Why? I'll tell you that later. B lymphocytes and some other killer cells, some other killer cells, they would call them, that reach to the site because the infection is now out of hand. So the blue one is the T lymphocyte, and there are many T lymphocytes in every tissue. Always remember, in every tissue we have many T lymphocytes. So when it would reach at the site of infection, now they would release signals, and those signals basically would call the B lymphocyte. The second thing that the T lymphocytes are going to do is they would scan the pathogen. Okay, now a few terminologies I have left. Now I need to explain those terminologies. First terminology that I need to explain over here is the pathogen. You know what is a pathogen?
Um, is it like a, what do you call it? Like a particle of virus?
Basically, anything that is causing a disease is called as a pathogen. So now the pathogen is can be bacteria, virus, and fungus. Secondly, there is another terminology called as antigen. What's an antigen?
Um, that's maybe a fighting against the disease.
No, no. An antigen is a molecule specific to a particular cell, to a particular cell. Suppose it works just like an ID card. When you go to school, most of the time what happens that most of the schools practice this particular thing, that they don't let the students enter into the school without showing them identity card. Is that right?
Yeah.
And if a person or a student tries to enter without showing their proper identity, they would not let them enter. And if they would try to enter, they would immediately neutralize them. So the antigen is basically a molecule that could be situated at any cell. Like our white blood cells, our own cells also have antigens. You should know. Like if suppose that this is my cell. Now this cell is having a particular antigen. This antigen has a particular quality, and that is it is recognized it is recognized as my own antigen and not be attacked, and will not be attacked. It would not be attacked if it is my cell and the antigen is present at my cell. So my body, my white blood cells would recognize it, and they would certainly defend it, and they would not attack it until there is a problem that is called as the autoimmune problem. Autoimmune problem is a thing in which your body's immune system starts attacking your own cells. So that is basically the autoimmune problem.
So is it
If the
autoimmune disease?
Yeah, autoimmune disease, yes. So in case of autoimmune disease, your antigens basically your immune system basically recognize your antigens as the enemy antigens. So they would start killing your own body cells. But uh, in the normal immune system, your body would recognize your immune system as the friends, and it would not attack them. Let me give you a very strange idea. Your white blood cells would attack the very sperms that would enter into the female body. They would immediately kill them. Why? Because those are cells and they have different antigens. So it is strict to that level. It has a rule up to that level that even if a secretion from a human body is entering into another body, it would be rejected. The cell that goes to the egg is one of those cells which has successfully deceived your immune system. That cell would only be able to reach to the egg at the time of fertilization. So it is strict to that level. So your immune system basically is would search for the antigen. Antigen. In that sense, basically there are two types of antigens. One antigen is called as self antigen, and the other one is called as the non-self antigen. So in this case, the site of infection, in this case, this site of infection is basically filled with non-self antigens. This site of infection would basically be filled with non-self antigens. Okay, now uh, there are antigen receptors, which are located at T lymphocytes and B lymphocytes. Now, focus. T lymphocytes, when it comes to T lymphocytes, I drew the T lymphocyte in blue on the previous slide, so I'll do the same. This is a T lymphocyte. Now, the T lymphocyte's receptor would never leave it. Its receptor would stay on the cell membrane. This is the cell membrane, and this receptor would stay on the cell membrane. But what do the B lymphocytes do? B lymphocytes have another technique. They would be having the antigen receptor on their cell membrane, but they would also release these receptors into the blood. They would also release these receptors, which means that basically these receptors are no more limited to the cell membrane, but they are being released into the blood. They are being released into the blood, and once they are being released into the blood, it would be these would now be called as antibodies. So antibodies are nothing but those receptors which were supposed to be sticking to the cell membrane, but the B lymphocytes are going to release these receptors into the blood. Is it clear?
Yeah.
Now, there is another very important property of these receptors, that they are complementary to antigens. Complementary to antigens. Which means that only that receptor or antibody, only that receptor or antibody would attach to antigen which is complementary, which is complementary. Otherwise, it would not attach. Suppose that there are this is the infection site. The T lymphocyte having the receptor would reach there. There are three different T lymphocytes having three different receptors. Now, all these three would go to the infection site, all of them. But over here, remember I'm writing one over here, only that lymphocyte could capture antigen, antigen of pathogen, antigen of pathogen that is complementary. Other T cells would be rejected. Other T cells would not be able to attach to it. So what does it typically means? It typically means that whenever your T cells or B cells are going to the infection site, only those T or B would combine to the pathogens which are actually what? Which are complementary to the antigens present on the pathogen. Is it clear?
Yeah.
Okay. Now the what now the T lymphocyte is going to do? So I was basically mentioning the step-by-step procedure. So first I mentioned this as one. So I'll change the this. I will say this A. So this point would be mentioned over here as A. If you come back to the Jamboard, remember this A represent additional point. Okay. Uh, the
Okay.
step-by-step procedure I am referring with the numerics. So first phagocyte would go. Second, the T lymphocyte would go. Third procedure, the T lymphocyte would release signals. Fourth procedure, the T lymphocyte would scan the pathogen, copy the antigens and produce its clones, which would be called as memory cells in this case. Basically, what's going to happen? It's just like it's just like suppose I am uh, suppose I am basically I am basically I basically go to I find a document. I I find a document, and that document is very important. So what would I do? I would copy it and I would try distributing it to the others. In
Yeah.
modern form, I would take the picture. I would take a picture of it, and I would try to send that picture to as more people as I could, as much people as I could. Similarly, what this T lymphocyte is doing, it is scanning in the fourth procedure, it is scanning the pathogen, copying the antigen, and produces its clones. It is going to produce more copies of clones containing containing complementary receptors. Now the T lymphocyte is basically uh, copying itself. It is producing more clones, but are only having these clones are only having complementary receptors. What would it mean? It means that whenever we find an antigen, our body assumes, please focus on the sentence, I would write it over here. Whenever we face an antigen, our body assumes that this would, I'm talking about non-self antigen. Our body assumes that this would come back again. So body would make many copies, or clones of those cells which are containing complementary receptors, complementary receptors to the antigen, to that antigen. And these clones are basically called as memory cells. Did you understand this?
Yes. Yeah, I
Basically, our
understand.
body, yeah, sorry. Okay.
I understand. Yeah.
Basically, basically, our body is preparing itself for another attack. That if it can enter into the body, it means that it is prevalent and it would enter again. So now, when the T lymphocyte would scan it, it would immediately clone itself because that T lymphocyte is complementary to the antigen. Now it is prepared. But the thing is, the story does not end here. The story does not end here because these T lymphocytes are calling the B cells. So B cells would now go to the infection site. They would now go to the infection site. This was fourth step. Now this is fifth step. They would now go to the infection site. After reaching, the B cells would do two things. This is sixth step now. The B cells would do two things. Number one, they would create more memory cells. Now that you know about the memory cells, so I don't need to explain it. Is the memory cell clear?
Yes.
So uh, now the second thing that the B lymphocytes do, they would now release some more cells which are called as plasma cells. And the plasma cells are those which would release antibodies antibodies into the blood. They would release antibodies into the blood. Remember that the plasma cells are B cells. Just their function is a bit different. The B lymphocyte is going to the site, scanning the antigen, producing the memory cells and the plasma cells. The plasma cells are just a copy of B cells that are performing the function of releasing the antibodies into the blood. These antibodies, what is the function of an antibody now? We need to know that. The antibody arrests the pathogen by attaching to its antigen. By attaching to its antigen, and can also act as a signal for phagocytes. It's just like you have caught a person, but you don't have authority to kill it. You go to the you go to the site. You go and go to the you can say police station, and the police would do the rest. So it means that you are basically an antibody which is seizing the pathogen, arresting the pathogen, and then it would just simply act as a signal for the it would act as a signal for the uh, phagocytes, and when it would done acting as a signal for phagocyte, the phagocyte would come and kill it. I would suggest you to uh, within 2 to 3 minutes revise the topic. You got the link for this Jamboard?
Yeah.
Okay.
Um. Oh yeah. Now it's refreshed. Yeah. Okay. Yeah, I have it. Okay, done.
Understood?
Yep.
Okay. So basically, this kind of reaction of immune system, this is called as primary response. A primary response means the primary response means that uh, this type of response would happen would happen when the would happen in every disease. It would happen in every disease, and it means that you basically are going to respond to every antigen that would enter into your body. That is the primary response. That is the primary response. And the primary response always takes time. It always takes time. What does it mean? It basically means that the primary response could take from 4 to 10 days. 4 to 10 days. And the 4 to 10 days means that uh, depending on the organism, on the organism, your immune system and the health of your immune system, the disease, the cure of the disease or tackling the disease would take time. Tackling the disease would take time. So uh, within in 4 to 10 days, what is going to happen? Number one, the memory cells would be formed, and antibodies would also be synthesized. But but the thing is, that when we draw a graph, when we draw a graph, uh, initially, okay, number and over here time. If we draw the graph initially from the zero, day one, day two, day three, day four, day five, day six, day seven, day eight. Now, suppose the red line is showing the pathogen. So from the almost first day, the pathogen number of pathogen would rise. Number of pathogen would rise. And from now, these are the pathogens. This graph represents the pathogens. From almost 4 to 5th day, the antibodies would start synthesizing, would be synthesized, and the number of antibodies would just take a very steep rise, which would lead to a fall in the pathogens right at the time when the antibodies are being created. And by the end of the graph, the pathogens are totally finished and antibody number is still on the rise. It is still on the rise. So, but you should know that this 8-day period is the period of sickness almost. And this variates. This is variable. It does not mean that in every disease it would happen. It is variable, but it happens like this. And then now this is a primary response. But in the secondary response, there is a secondary response. In the secondary response, what happens that the same organism, same antigen enters the body, enters the body. Now the graph would look different. This is the graph that I drew over here. This is for primary response. Now your body is already prepared. Your body has memory cells, and remember that memory cells can live up to 20 years. Now the graph would look like this. Over here, you would write the number of antibodies or the pathogens. This is time 0, 1, 2, 3, 4, 5, 6, 7, and 8. And this graph is now representing the bacteria. Within 1 to 1.5 days, the bacterial graph would rise and fall, or maybe in 2 days. But from the very first day, the antibody graph is rising. This is the graph for antibodies. And why it happened? Because the memory cells were prepared. Memory cells were prepared. Now the memory cells, let me give you another explanation. These memory cells, the memory cells of B lymphocytes, upon the second exposure, you know what would they do? Upon the second exposure, the memory cells would do two things. They would turn into plasma cells, and they would turn into more memory cells. Now, can you tell me what is the benefit of increasing the memory cells on the second exposure?
Um, so that if there's a third exposure, there'll be more memory cells.
There would be more. It basically means your immunity is getting strengthened. Your immunity is getting strengthened. And what is the purpose of plasma cells?
To release the antibodies into the blood.
They release the antibodies. Plasma cells could be removed from the blood after 6 months or 1 year. The plasma cells could be removed from our body, but memory cells are they live up to 15 to 20 years, and that would be very much beneficial because upon the second exposure or secondary exposure, your immune system is ready and it would immediately kill the same organism, same antigen. So that is how it would work. This is the graph for secondary response. And secondary response, it is called as secondary response due to reason that it does not happen in case of all diseases. But primary response happens in case of all diseases, all types of antigens, whatever enter into the body. Okay, now the immunity, most of the time we say that on the basis of involvement of body, involvement of body. Oh, okay, no, sorry. This topic is separate. I'll tell you that later. There is a phenomena in which we inject antigens into our bodies, externally. What we call that phenomena?
Vaccine.
Yes, that phenomena of injection is called as vaccination, and that solution is called as a vaccine. Basically, the antigens can be injected in three forms. You can say forms of antigens. First is inject the antigen by injecting the whole dead pathogen of same disease, of same disease to which you want to get vaccine. You kill that organism and you inject that killed organisms into the inject those killed organisms into the body. Second is the pathogen is alive but weakened. Like you have removed pathogenicity, removed their virulence, or you have removed their pathogenicity.
Your voice is cutting.
Is it fine now?
Yeah.
Hmm. We remove the pathogenicity, we treat that organism, we remove its virulence or pathogenicity, but we do not kill it. That is the second form of that is the second form of vaccine. First form is you kill the organism and then you just inject it into the body. That organism, that killed organism, would act as antigen. Second, you treat that organism, weaken it, and then you inject it into our body. And the third thing is you uh, inject only antigen. You inject only antigen. Antigen is a very small molecule. You do not inject the pathogen, whether it is dead or alive. You do not inject the pathogen. You just inject the antigen, very small molecule, into the body. All of these would result in provoking the body. All of these all would result in provoking the body to show primary immune response. Immune response. So this thing is called as vaccine. Okay, so primary immune response. The primary immune response would lead to, uh, you can say, immunity. It would lead us to immunity. But have you ever thought about the second dose of vaccine?
In what sense?
Like, why did we get the second dose in Corona?
Because the disease evolved.
Hmm. After studying about the secondary exposure, what do you think would be the benefit of the second dose or the booster dose?
Add more memory cells.
Yes. Every time you take a dose, basically you are increasing your memory cells. So it means that whenever you are doing the vaccination, you are increasing your memory cells, which means the chances of getting exposed and the chances of getting infected from the same organisms again and again would reduce. It would only reduce, it would never be eliminated. Okay, so next is, now these are the types, and always remember the vaccine is made uh, according to prevalence of the diseases in our area, in a particular area. So it does not mean that if you are in UK, you would certainly be getting a vaccine against the disease that is prevalent in Pakistan. Or if I am living in Pakistan, I would be getting the vaccine that is prevalent in UK. Basically, it depends upon the area. Okay, I like you to revise it. I have to go. I have to go to the loop. So I like you to revise it. I'm just coming back.